Welcome, everyone. I have to take my reading glasses off to see all of you, but it's truly wonderful to see so many of you here in person today. And my understanding is we are joined by at least several hundred people who have signed up to see this lecture virtually. So a wonderful turnout, I have to say, Dr. Roth and Dr. Barondes, that this is very unusual right now post-pandemic. So a tribute to the interest in the lectureship and in today's lecture. So I want to welcome you, and I have the great pleasure today in a moment to introduce our speaker, Dr. Bryan Roth. But before I do, as I do every year, I want to take a moment to reflect on the lectureship and on the contribution of its namesake, Dr. Barondes. He is a man who helped found the scientific discipline of neuroscience, a pioneer in elaborating the biological basis of mental illnesses, and of course, someone who's had an enormous and lasting impact on us here at UCSF. Sam is chair of the Department of Psychiatry in 1986, saw the future of the field, and was determined to build it here. He set his sights on identifying and recruiting the very best young physician scientists in the country, betting on molecular biology and human genetics to provide the key insights into the causes of psychiatric illnesses as a necessary step to developing better treatments and ultimately cures. It's difficult to overstate, I think, just how prescient this vision was and is. It is as important and vital today as it was 27 years ago. As current chair of the Department of Psychiatry and Behavioral Sciences, it is a remarkable, I think, but true statement that a central goal of mine since coming here in 2013 has simply been to execute on the path forward that Sam articulated nearly 3 decades ago. I want to add that his lasting impact goes beyond his discoveries at the bench, his articles, his lectures, and his wonderful books. He's advanced the field with an unparalleled eye for talent, and remarkable persuasiveness. The physician scientists that he managed, often with very modest means, to bring here to UCSF over the last 2 decades is truly a who's who of neuroscience and genetics. Whenever I talk with Sam about his career, though, at UCSF, in addition to all of these achievements, he always mentions his leadership of the Arts Committee. True? He's had a hand in beautifying our campuses, both here in Mission Bay and at Parnassus. When we think about it all together, his discovery of galactans, transforming contemporary psychiatry, spotting and nurturing the brain trust of several generations in our field, Sam has made the broadest possible mark on our community. It's an honor and a pleasure to host this lectureship today to celebrate and recognize his many accomplishments. And I also have the great pleasure of welcoming Dr. Bryan Roth, MD, the Michael Hooker Distinguished Professor at the University of North Carolina. Dr. Roth completed his medical school at St. Louis University, was at NIH, did a psychiatry residency. Is this the right order? Did a psychiatry residency at Stanford. It's located about 30 miles south in Palo Alto. And then he completed his fellowship at the Nancy Pritzker Laboratory in 1991 there at Stanford. He started as an assistant professor in the Department of Psychiatry at Case Western Reserve in the School of Medicine, where he ultimately became professor of biochemistry with secondary appointments in psychiatry, oncology, and neurosciences. I think even in the title reflect a reflection of the tremendously broad impact that he's had on the field, on science in general. In 2006, he moved to UNC Chapel Hill to become a professor in pharmacology with a joint appointment in chemical biology and medicinal chemistry, and director of the program in translational proteomics. And then in 2007, he was awarded the Michael Hooker Distinguished Professorship in protein therapeutics and translational proteomics. From the outset, Dr. Roth's laboratory has made truly groundbreaking discoveries in molecular pharmacology and synthetic biology. He's been a leader in elaborating the structure and function of the serotonin and opioid receptors, among others. He's led the field in developing novel cell-based screening platforms to discover tools to study G-protein coupled receptors, and he's pioneered large-scale chemical genetic approaches to discover, test, and validate novel molecular targets for therapeutic drug discovery. He invented the chemogenetic tool called, ironically, I think, DREADS. Did you consider DELIGHTS as an acronym? Sorry. Anyway, little. Which DREADS? Yes. But regardless of the name, a tremendously important tool in gaining traction on studying circuit level behavior, both complex and simple, and clearly a tremendously consequential tool in moving the field of psychiatry and psychiatry science forward. He's had an enormous impact in the field reflected by any measure, an H index of 141, more than 87,000 citations, and recognition by Thomson Reuters as a highly cited scientist over so many years I stopped counting, but this is in biology and biochemistry and pharmacology. He's one of the few psychiatric physician scientists, although sitting next to one, who has been elected to both the National Academy of Medicine and National Academy of Science. It is humbling to review even a short version of your CV, and elaborating his publications in just the journals Cell, Nature, and Science would take up the rest of our time together. So I would just say that Dr. Roth is the quintessential psychiatric physician scientist and truly the epitome of what Sam Barondes envisioned for the field and supported throughout his entire career. It's difficult to think of a more fitting person to deliver this named lectureship. Welcome, Bryan.
Wow, that was really nice. Thank you so much. That's one of the nicest ones I've ever had. When I get these, I always think it would have been great for my parents to hear that. You know, sadly, they never did. Before I start, I just want to say a few things about Sam. So he is and was an amazing chair. He recruited the most outstanding basic scientists in psychiatry. And I'll just give you a couple of anecdotes here off the cuff, because I didn't have any prepared remarks. But Sam interviewed me for UCSF back in it was a long time ago. And he said to me, he considered Stanford University his AAA farm club, basically, which was, I thought, very interesting, but true, actually. So he recruited John, recruited Mark von Zostro, Rob Malenka, right? And they, you know, I don't think there was a single dud in any of the people you recruited, which says a lot, actually, particularly in psychiatry. Sam was very, even though I was, you know, I didn't come here, basically because I didn't think I would make it. I mean, Sam was extremely persuasive, you know, came up with an offer which was highly competitive. But in that era, it was like 2 years, you get 2 years funding, and then, you know, you have to get money from the NIH. And I was actually worried, rightly so, that it would take me longer than 2 years to get funding. And it did. It took me a long time to get funding. So if I came here, I would not have survived. I would have died. So it was actually a good thing that I didn't come here. Maybe. I mean, you'll see from the talk today, if you agree. The other thing about Sam was that of all the people, all the sort of senior scientists in psychiatry, I would say he has been, in my life, the most personable and friendly and supportive to me. Every time I see him at an ACNP meeting, he's always the first to come up and greet me, ask me how I am, always interested in my research. He's one of the few people that actually came to some of my talks at the ACNP. And I remember after one, it was really early on in my career, I was having a very hard time getting funded. It's sort of the foundation for a lot of the stuff that I do now. And it was trying to find out what is the mechanism of drug action in the brain. That's what I was really interested in. And at that time, it was trying to find out what receptors drugs bind to, which was more or less unknown at that time. And of course, nobody had any interest in that. NIH certainly wasn't going to fund it. But after my talk, Sam came up to me and he said, continue this. He said, this is really important. And that stuck with me for a long time. So thank you. Thank you very much. So what I'm going to talk about today are psychedelics. This is a talk in 3 parts. So the first part, I'm going to just talk about what is known about psychedelics, the role of various serotonin receptors, then present some new findings from my lab on the molecular details of psychedelic drug action, and then some new things that I think are really exciting. So we'll see. So I guess you probably have heard, people here have heard of psychedelic drugs before. Yes, most people have. So what is a psychedelic drug? Well, I use this definition of psychedelic drugs, which is a drug which has an LSD-like effect in humans via activating the 5-HT2A serotonin receptor. And this is a definition which is very similar to the definitions of the FDA, the Drug Enforcement Agency, and the EU Medicines Consortium, okay? But if you read almost any scientific paper or the newspaper, this is not what a psychedelic drug is. Instead, psychedelic drugs are really basically every drug that is a hallucinogen that we would consider hallucinogenic is considered by most scientists and certainly most non-scientists to be quote unquote psychedelic, okay? And this has caused a lot of confusion in the field. So what I'm gonna do is I'm gonna spend a couple of minutes going over how I classify these drugs and how these drugs have been classified by everyone else who studied these drugs until about 2 years ago, when newspaper reporters got interested in the field, okay? So they're hallucinogens, they're psychedelics. These include drugs like LSD, psilocybin, and mescaline. These are all natural products or semi-synthetic. Then we have dissociative agents, and these are drugs like ketamine and PCP. So technically, these are dissociative anesthetic agents. They're not psychedelics. There are compounds that induce delirium. And for those of you who are physicians, you know that delirium is different from a psychedelic experience. And these are primarily anticholinergic agents. And then we have this interesting class of compounds called oneirogens. So oneirogens are drugs that induce a dreamlike state. And this is a different state from that induced by psychedelics. And one of the advantages, so I'm going to talk about these drugs here today. And one of the advantages of this type of classification is that it's dependent on the mechanism of action of the drugs. Okay, so psychedelics are 5-HT2A antagonists. These drugs are NMDA antagonists. These drugs are anticholinergic. And these drugs are kappa agonists, okay? And to prove to you that they cause distinct effects in humans, I'll show you a few videos. So this is an interesting, I got all these videos from YouTube. So this is an interesting video from the 1950s. So this is a woman who was given LSD in the 1950s. We don't know the dose, but you can see the experience here.
Well, tell me.
Well, I just couldn't. I couldn't possibly tell you. It's here. Can't you feel it? This whole room. This everything is in color, and I can feel the air. I can see it. I can see all the molecules. I'm part of it. Can't you see it?
I'm trying.
Oh, it's just like.
So at any rate, she's having these hallucinations, basically. She's seeing things. And the thing to notice about her is she seems sort of euphoric, right? A little bit euphoric, would you say? Yeah. So this is from somebody that has recently taken ketamine for antidepressant effects. Okay. This is 20 minutes of ketamine experience. And notice this guy.
Have you asked me anything? How's it going? How does it feel? Incredible. What are the thoughts that are in your mind? That we're all connected, that we're all together.
There's this sense of oneness that he has. Okay, I don't know if this is a common experience with ketamine, but people who classify ketamine as a psychedelic focus on this aspect of the experience. And last, my favorite, Salvinorin A. Okay, so this is a video I've shown before. Some of you have seen, but not everyone.
Yeah, I want to take an ashtray with Sally one time at Whitney's
My
house.
body's, that's what it is. My body's like numb. It feels good.
Cash that shit.
And hold it in. Don't breathe it out.
You feel it yet?
Nope, it's good.
Give me a thumbs up when you feel it.
He's gonna feel it about now.
My heart's going back.
Yeah.
Oh shit.
Now he's gonna start. Okay, so you get the idea. So each of these drugs cause a change in somebody's internal state, but they're different, okay? And my sense is that people who study these substances now are saying that anything that changes the internal state of a person is a psychedelic, okay? And you can use that definition, and if everybody uses that definition for a psychedelic, then eventually the definition will change in the dictionary. I'm just saying that there is another way of looking at these compounds that's based on science, and you might want to consider that. So that's all I'll say about that. But it's sadly something that comes up every time I give a talk. So I thought I would sort of short circuit it. But what are the reasons that psychedelic drugs have recently become sort of famous are based on these relatively small-scale clinical trials that were begun some years ago for treating depression with a single dose or 2 doses of psilocybin. And what was found was that a single dose of psilocybin or 2 doses of psilocybin separated by a few weeks has basically a very rapid and sustained effect on depression symptomatology in patients. And the effects are so robust that most patients are no longer depressed. And they're no longer depressed 1 year after they've taken this psilocybin dose and had a guided experience. So they become really interesting from a therapeutic perspective because they have the potential, and I think they will, completely transform how psychiatric care is delivered in the US, along with MDMA. So MDMA is technically not a psychedelic drug. It's what's called an entheogen. But MDMA has now shown 2 phase 3 clinical trials positive for post-traumatic stress disorder. I expect it will be approved by the FDA next year. And that's also going to dramatically change how we treat those sorts of disorders. And so with this as a background, my lab over the years has tried to understand how it is that psychedelics exert their action ultimately at the molecular and atomic level. And this is what I'm going to tell you about today. So I started studying actually psychedelic drugs and their receptors when I was in Costa's lab, and just continued it sort of as a side part of my lab because really until 2 or 3 years ago, there was no one in the world that was interested in what we were doing on psychedelics. I'm not sure that anybody is interested in what we do, but the topic is still interesting. And so when we began re-examining the actions of psychedelic drugs, the very first experiment we did was to try to understand what the molecular targets are for psychedelic drugs in the brain. And so I developed my lab developed a platform where the diagram of all of these and in red there, those are all the receptors that are activated by LSD at low concentrations. And of these receptors, there's only one that is responsible. That's the 5-HT2A receptor. But the first thing we realized was that these drugs have an extraordinarily complex pharmacology. But given that, you might ask, so what is the evidence that this single receptor in the brain is the site of action of psychedelics? And it goes back really to the 80s. There are foundational studies done by Richard Glennon's lab that showed that the psychedelic actions in mice correlate with their ability to bind to this receptor. And then there were studies done in knockout mice, one study done by Gonzalez Mazo et al., the other here. My lab in collaboration with Bryan lab, Mike Kaiser, showing 5-HT2A knockout mice do not respond to psychedelics. But the best data really comes from humans. And there are two sort of seminal human studies that were done by Franz Bullenweider's lab in Switzerland. And interestingly enough, in Switzerland, psychedelics were never made completely illegal. And there still were psychiatrists that were licensed to prescribe psychedelics for psychedelic psychotherapy. And among these is Franz Bullenweider, and he was, up until recently, the only person in the world that studied the effects of psychedelics in humans. And what he found was that the effects of both LSD and psilocybin were blocked completely by pre-administering a 5-HT2A selective antagonist, catanserin. Okay, so the best data we have that the effects of psychedelics are mediated by 5-HT2A receptors is this data here, where humans were given basically a blocker of the receptor, a drug that's very selective for it, and then we're given actually massive doses of LSD and psilocybin without no effect, basically. So, but are there other targets? So we began to study this, and here I'm getting into basically unpublished data. So we decided to study this in a real systematic way. So what we did was we went to the literature, primarily with the parallel as well as PCAL and TCAL from the Shulgin lab. And we identified around 240 psychedelic ligands of various chemical classes. They were then subdivided, and then exemplars from each class were then profiled against all the GPCRs in the human genome. We've also profiled them against all the kinases, and they don't interact with any kinase, so I'm not going to present any of that data. So we first did a radioligand binding screen, and this is the result. So those are the psychedelics across the top, mainly psychedelics. And given this, there's no way you would predict, given this heat map, that it's the 5-HT2A receptor here that's the site of action of all those compounds. No way you would ever predict that. We also did functional profiling. So as I mentioned, we've developed this platform where we can screen basic compounds basically against every druggable GPCR in the human genome. We can also quantify their signaling at every transducer. And this is the heat map of that data. So these are the compounds here. And then going up are the various receptors. And what you can see is this is a sparse matrix. So we have one compound here that's promiscuous, probably non-specific, but there are three basically areas of activity, and these correspond to serotonin, dopamine, and adrenergic receptors. And if we look at the transduction pathways downstream of these, so what we did is after we identified all these targets, we then interrogated them using assays that we developed to see if there was any signaling pathway downstream of any of these receptors that classified psychedelic drugs from non-psychedelic drugs. And there wasn't. There is none, okay? We know it's the 5-HT2A receptor, and we know it's GQ. There's no question about that, but there's no way you would ever figure that out based on this sort of information, because these compounds have very promiscuous pharmacology. So what we have done since then is we've made very selective compounds for the 5-HT2A receptor, and of course have tested them in mice. Now, despite this evidence, this sort of data, there have been sort of contrary things that have been published by various investigators in the past. And I'm just going to mention one of these. So this was a really interesting paper. This was published in Science some years ago. And they suggest there's this interesting compound, N,N-dimethyltryptamine, also known as DMT, which is an extremely potent, I would say powerful psychedelic drug. And they suggested, based on data in wild type and sigma-1 knockout mice, that the receptor really was the sigma receptor, not the 5-HT2A receptor. And the basis for this was that DMT caused a locomotor response in wild type but not sigma receptor knockout mice. Well, it turns out that we tested DMT for the sigma receptor, and we found that it had almost no affinity for the sigma receptor, 14 micromolar, okay? And around this time, I was working with Bryan Choiket's lab, this very talented student of his at the time, now associate professor here at UCSF, Michael Kaiser, who had built this chemoinformatic approach for predicting molecular targets for drugs. And to make a long story short, I asked them to put DMT into the similarity ensemble approach predictor, and it predicted, not surprisingly, that serotonin receptors would be the target of DMT. And so we tested this. We found that DMT is an extremely potent agonist for the 5-HT2A receptor. Its affinity for the 5-HT2A receptor is about 100 times higher than its affinity for the sigma receptor. And if you look at knockout mice for the 5-HT2A receptor, they do not respond in sort of the prototypical way, which is the head twitch response. So this is, and it also has 100 nanomolar affinity. So there are some other of these sorts of studies that have appeared throughout the years, but by and large, I would say the evidence is pretty overwhelming that the 5-HT2A receptor, this is the receptor for psychedelic drugs. Okay, so given this, what is our current understanding of psychedelic drug action? And this data comes initially mainly from my lab. So what we found many years ago was that 5-HT2A receptors are localized mainly to layer 4 and layer 5 cortical pyramidal neurons in both humans and rodents. That downstream of the receptor, this is a GQ-coupled receptor. As I'll show you, basically all the actions of psychedelics are mediated through activating GQ and associated signaling pathways. And that this as well is something we discovered along with Peter Penzis in 2000, I'd like to point out in 2009, that psychedelic drugs induce plasticity in neurons in culture. And they do this within 30 minutes. And it's thought that this effect on plasticity is related in some way to the therapeutic actions of psychedelic drugs. Although I would say it's currently not entirely clear how that might be. So the first thing that we wanted to understand, since the 5-HT2A receptor is the target of psychedelic drugs, is how is it that drugs bind to this receptor, okay? And this as well is something I studied for many, many years, sort of in obscurity. So this is our first molecular model of LSD there in blue versus ergotamine binding to the 5-HT2A receptor, published in the venerable journal Molecular Pharmacology. And starting in around 2007, we began to solve structures of various GPCRs with the ultimate goal of solving the structure of the 5-HT2A receptor in complex with LSD, which we finally did. And these studies were initiated in collaboration with Ray Stevens and Vadim Sherzov, and with Eric Xu and with my own lab. So over the years, we solved the structures of all the opiate receptors, smoothen receptors, melatonin receptors, all the serotonin receptors, all the dopamine receptors, and all the this interesting family of receptors called MRG family of receptors. So we're able to solve most of these initially by lipidic cubic phase, X-ray crystallography, and more recently by cryo-EM. And I'd just like to point out two recent papers. This, for those of you who use chemogenetics, I urge you to read this paper. I think it's a really important paper on why chemogenetics actually works. And this is sort of a nice legacy paper where the structures of all the opiate receptors were solved with their endogenous ligands in a single paper. So it's a nice bit of archival work. So we also ultimately solved the structure of the 5-HT2A, there are three members of this family, the 5-HT2B and the 5-HT2C receptors, initially by X-ray crystallography and more recently by cryo-EM, bound to psychedelic and non-psychedelic drugs. And these findings have more or less all been published. And rather than taking you through many structural biology slides, I have this nice little video, courtesy of the TV show Hamilton's Pharmacopoeia. So what they did was they took our structural information and made this nice animation here, which I'm going to show you. So this is LSD here. Firing. Okay. So we also solved structures with arrestin. And this was by we, I mean my lab in collaboration with Yorgos Skiniotis' lab. This is, for those of you that do structural biology, I think this is like a really, really good paper. We got the APO receptor, we got the GQ coupled complex, and we have the beta-arrestin 1 complex, all in the same paper, all bound to LSD. So it's a nice, thorough paper. So in the time remaining, I want to talk about some new findings here. And so one of the things one of my postdocs has been doing, Ryan Gumper, is to basically solve the structure of representative psychedelic and non-psychedelic drugs at the 5-HT2A receptor. So we have all these structures here, pretty good structures. And the idea behind getting all these structures has been, if we get a structure of the receptor, then that will be helpful for us if we want to find new drugs that interact with the receptor. And this is basically the idea. And as we were solving these structures, the AlphaFold paper came out. I remember when it came, the day after it came out, there was an article on the front page of the New York Times, which my wife forwarded to me, and she said, "You don't have to do any more structural biology. They've now solved all the structures for you." So I said, "Thank you. That's a wonderful thing to hear." So this is the AlphaFold structure prediction for the 5-HT2A receptor here. And we also solved the structure of it in complex with a non-psychedelic drug, glyceride. This was done by Yorgos' lab. And if you look at the binding pocket in the structure, in the real structure, it's the drug sort of fits in there pretty tightly. But if you overlay the AlphaFold structure, the side chains are now starting to intrude into the binding pocket. And so you actually cannot dock glyceride at the AlphaFold structure for the 5-HT2A receptor. In fact, you cannot dock any known compound at the AlphaFold structure, okay? So given that, I proposed this experiment, or we thought about it simultaneously, I think, with Bryan Choiquet. And in many ways, this is the most exciting and the most disturbing experiment I've done. And so here was the idea. So we have this AlphaFold prediction, which is wrong, as far as we know. And we have the structure, which we think is right? And so if we use both structures for drug discovery, which works better, okay? So in other words, can AlphaFold provide a template for drug discovery? So this was tackled by JK, a former postdoc at Bryan's lab, now with his own lab at Rockefeller. And so here was the experiment. So we have the structure. So using ultra large scale docking, 1.6 billion molecules were docked against the structure of the receptor. And then ultimately they were ranked, and 223 were physically tested, okay? And we did the exact same experiment with the model, 1.6 billion compounds, same compounds, 165 tested, okay? And both approaches had identical hit rates of about 20%. But if you look at our best compounds, these are our 3 best compounds. And you can see that this compound here is the best. So these came right out of the docking screen, no further chemical optimization. This compound has a 30 nanomolar potency at the 5-HT2A receptor and is absolutely selective. So this is better than any known psychedelic drug in terms of potency and selectivity. And this came directly out of the AlphaFold, okay? In fact, all 3 of our best compounds came out of the AlphaFold study, okay? So here's what we think is going on. So when there are now ways to basically reanalyze or further analyze or interrogate the structural information that you get from cryo-EM. And for those of you who do that sort of work, you know that the structure that you get is not the structure. It's basically an average structure of probably hundreds of intermediate states that are captured when you freeze the proteins and put them on the plate. But there are ways now to pull out those other structures that are there. Using CryoSpark or other programs. And so what Bryan did was went back to his data. Okay. He went back to his data And I just want to point out two compounds here, LSD and BOL. BOL is bromo LSD. It differs from LSD in a single atom. It's a bromine. Bromo LSD is not psychoactive, okay? You can take a gram of bromo LSD and nothing will happen except you'll have a headache, okay? LSD, by contrast, you take 100 micrograms of LSD and you will have a very intense psychedelic experience, okay? So in deconstructing these intermediate states, we found 123 intermediate structures for each ligand. They were then mapped by principal component analysis. And what you can see is BOL is completely distinct from LSD in the ensemble of states that it stabilizes, okay? And we basically think that what's going on here with AlphaFold is that AlphaFold is picking up conformational states that are not captured by our physical means, either cryo-EM or X-ray crystallography, but that are potentially useful. And one of the nice things about the AlphaFold versus structure was that when we looked at the chemical structures of the individual molecules that were predicted by either the model or the structure, the universe of chemical compounds was different. And so what the advantage actually then of doing these computational studies on predicted structures versus real structures is you basically double the amount of chemical space that you end up ultimately being able to interrogate. Okay? So it may not be completely useless to solve structures. The last thing I want to mention here is, okay, so we have about 15 minutes, is recent work on how psychedelics exert their actions in vivo. We decided again to basically take an unbiased approach and start from anew. There were no antibodies that are useful for studying them. So what we did is we being Justin English, designed basically a mouse that we could prepare where we've tagged the receptor with GFP. And I'll just to make things quick, we tagged it in such a way that we don't affect anything we can measure with respect to the protein. It still signals the same. It's targeted and trafficked and regulated identically to the wild type receptor. And mice that express the GFP tagged 5-HT2A receptor respond to psychedelics the same as wild type. Okay, so we made the GFP tagged receptor. We put in an inducible CRE downstream of that. This turned out to be extremely important because if you use a constitutive CRE, there's transient expression of 5-HT2A receptors during development, and every cortical neuron gets labeled. Okay, so it took a bit of work to come up with this approach, but now you see this beautiful expression of 5-HT2A receptors there in the cortex, as well as in the striatum. And for those of you who study drugs of abuse, those patches co-localize perfectly with mu receptors, okay? And then we did whole brain mapping by light sheet microscopy of cleared brains. And this is basically the complete map of the receptor. So you know, it's mainly in the cortex. There's a little bit in the striatum, some occasional cells in the hippocampus. And when you look at the cells in the cortex, 99% of them are pyramidal neurons. There are occasional fast spiking interneurons, parv neurons, but very few that express 5-HT2A receptors, almost exclusively expressed in these pyramidal neurons. And in particular, they're expressed in one subdomain, the apical dendrite of the pyramidal neurons. You can see this very well in the confocal micrographs. And we believe that this is the site of action of psychedelics. So one of the things that's interesting about these layer 5 pyramidal neurons is that they integrate information from multiple cortical regions and ultimately give us a view of reality that we call reality. Now, what psychedelics do is, because of their localization to the apical dendrites, they basically cause asynchronous firing. That's actually a fairly small effect. I'll show you this in just a minute. So that there is noise that's injected into the very neurons and into the very parts of the neurons that are responsible for giving us our perspective on reality. And so the psychedelic experience is just, I think it's probably just noise. That's it. And the brain is very good at making meaning out of nothing. In fact, one can speculate that the reason we have the brain is to predict things that don't exist. And so it just makes up a story about what's going on. And for reasons that are not entirely clear to me, this story that's made up when people take psychedelics is very meaningful to them. So it's instructive to go back and look at Roland Griffith's studies on depressed individuals, because he interviewed these people that participated in the psilocybin trials a year later and asked them to rate the top 3 experiences they had ever had in their life. And uniformly, they rated the psychedelic experience as one of the top 3. Okay? Right up there with the birth of their child, getting married, getting divorced, etc. So for reasons that aren't entirely clear, activating this receptor makes you think everything is significant. Now, I mentioned that we think this is all due to G-alpha-Q activation. I'll just present the data for this. This is all unpublished data from a very talented postdoc of mine, or student of mine, Gavin Schmitz. And so because we had these mice, we're now able to cross them with a reporter line that expresses TD tomato. And then we can put an electrode in a slice, in a brain slice directly on a 5-HT2A expressing neuron. So we know we're recording from 5-HT2A neurons. And when psilocybin or psilocin is applied, there's this increase in firing. You can see here, it's pretty small, but it's quite reproducible. And this firing is blocked 100% by a highly selective inhibitor of G-alpha-Q, this FR900-359 we got from Evie Castenis, completely abolishes the firing. And this is also blocked by 5-HT2A antagonists. So we know it's completely due to 5-HT2A receptors through G-alpha-Q signaling. As I mentioned, after there's this increase in firing, there can be spines that are formed. This is something we discovered many years ago. This is a collaboration with Peter Penzis. And we found that within 30 minutes of exposure of cortical neurons in culture, there was an increase in spine formation. And of course, this has been now replicated by many dozens of labs. There are a couple of very nice papers by David Olson's lab, also by Alex Kwan's lab. And recently there was this interesting paper by the David Olson suggesting that the effect of psychedelics might be through intracellular 5-HT2A receptors rather than surface receptors. I don't know if this is true. And certainly there are some parts of that paper that are ambiguous. But I'd just like to point out that over the years, my lab has studied the targeting and trafficking of these receptors. And they're, not surprisingly, a lot of them are inside the cell, in complex with arrestins, in complex with cytoskeletal proteins. And they're also found at the synapse. So it's not, if you read his paper, he says 90% of the receptors are inside the cell. This is not true. This is based on studies that were done with an antibody that's no good. But there are certainly some receptors inside the cell. Some of these are found in intracellular parts. Others are, a lot of them are actually found at postsynaptic densities. Okay. So as you can see, we're sort of going down each step. So we found the receptor, we found how the drugs bind to the receptor, we found out how they signal through the receptor. And then, so what is the next step? Okay. So this is the way we're going. So again, there was nothing we could rely on. And so we decided to do something super ambitious, thanks to money we got from DARPA, which is to do proximity proteomics in vivo. And so what we did, and I didn't think this experiment would work, but as I'll show you, it worked like gangbusters. So what we did was we tested a number of proximity ligation enzymes, and Turbo ID, for reasons that are not entirely clear, worked best with this receptor. So we made we put Turbo ID on it. We tested it in vitro. We also did it in the background of a humanized mouse. So it turns out that the mouse receptor differs from the human receptor in a single amino acid. And this single point mutation makes a huge difference in psychedelic pharmacology. So we made the mice, and then we just injected them with biotin. So this is a biotin ligase. And, you can see on the Western blots probed with streptavidin, there definitely is biotinylation. Okay, so that was great. But this was the thing that really blew me away. So this is in red. That's the staining pattern with streptavidin 594. And you can see it's essentially identical to the staining pattern of the 5-HT2A receptor. So not only did proximity labeling work, it worked in a very spatially localized way in the brain, which I was really surprised about, that it would actually work this nicely. And then, of course, once you have proteins biotinylated, you can do mass spec. And so in collaboration with Scott Soderling's lab, we did a control experiments where basically Turbo ID was expressed in every cell in the cortex. That's the control. And then the contrast is soluble Turbo ID expressed in every cell solubly versus 5-HT2A receptor Turbo ID. And you can see on the volcano plot, there were a huge number of proteins that were differentially expressed. As you might guess, if you compare the proteins that are expressed in pyramidal neurons versus all the proteins that are expressed in the cortex, this should be a completely different universe of proteins. And so we focused on this here. And the most relevant finding here is the 5-HT2A receptor, 64-fold enriched. So when we saw that, we knew the experiment worked, okay? And then, of course, there are proteins that are known to be downstream of 5-HT2A receptors, PIP2, GQ, beta-arrestin, PSD95, all enriched. Interestingly enough, NMDA receptors also in this complex, along with a lot of postsynaptic density proteins. And some of these are phosphorylated after psilocybin administration, which is pretty interesting. So if we take a step back here, these are the sort of proteins. So in green, these are known interacting proteins. That's the 5-HT2A receptor. Then we have actually a lot of proteins are enriched that are involved in axon guidance and synapse formation. So we think that this is probably a complex that's formed where new synapses need to be made. And then there's one protein there, N-acetyltransferase. This is for protein elongation when proteins are being made in the rough ER. So we think these are probably intracellular receptors, but most of these actually are proteins that are found on the cell surface. So we don't see a lot of evidence that, as was suggested, these receptors are primarily intracellular. We find they're actually mainly on the cell surface, and the proteins that interact with them are known to be on the cell surface. So that's where we are, and where are we going in the future? So we've really gotten interested in these complexes here that you can see. And we think that that actually there might be a very large multi-protein complex that is important for mediating the effects of this really interesting receptor. And so we're developing technologies now where we can isolate these synaptic complexes intact And then we're attempting to solve their structures by cryo-EM and by cryo-electron tomography. So we're really interested in these complexes. The other thing we're really interested in are these alternative states of the receptor, arrestin bound, G protein bound, intermediate states, starting to do time resolved cryo-EM to get some handle on these. And then very important and valuable contributions with the Choiquet lab. The idea is once we get these intermediate states, we can then use them for docking experiments. And the idea is that we'll get this completely different universe of compounds. And then we can go back and solve the structures and see if we can actually capture these with the new molecule. So that's where we're headed. So thank you very much. I just want to point out all this work is funded by your tax dollars. So thank you. And then I've been blessed with this really outstanding group of postdocs and occasional graduate students from my lab who spend a few years in my lab and then go off to do their own thing. I just want to focus on a couple here. Dan Wacker brought structural biology to my lab from Ray Stevens' lab. I'm eternally grateful to him. Shang solved the first LSD structure. And then Ku solved the structure of the complexes along with Chan here. And then Jonathan Fay recently has been helping us out with cryo-EM, the collaborations with Wetzel and Soderling's lab at Duke. Of course, my long-suffering and long-standing collaborator, Bryan Choiquet, and Yorgos Skiniotis really helped us out a lot to learn initial things about cryo-EM, and of course, Dave Nichols. And that is it. I'm happy to answer any questions you might have. So thank you.
When in the audience has a question, just raise your hand and I'll bring the microphone to you.
Great talk, thanks. So I was just wondering why, in your definition of psychedelic, specifically the drug LSD is named. Like it could be any drug, in theory. I don't actually even disagree. I'm just curious.
Because the term psychedelic was coined by somebody who took LSD. So I think his last name is Humphreys. I have a reference to this. So he published a paper in the 50s, early 50s, where he coined the term psychedelic. And he himself had taken most of these drugs that I list here. He hadn't taken ketamine because it wasn't known, but he took belladonna. He had taken ibogaine. Where he got it, I don't know, and LSD and mescaline. And he recognized that the effects of mescaline and LSD were distinct from these other drugs. And so he basically coined this term psychedelic for a drug that causes an LSD-like experience. Because at the time, LSD was before psilocybin had been discovered by Westerners. Okay? That was not discovered until the 60s. And so that's why we use LSD as a comparator, just because of a historical accident. But you could use any of these compounds. There's a recent paper published by the Swiss group where they gave human volunteers psilocybin or LSD in a blinded fashion, and then asked them to distinguish between the two, and they could not. Okay. 12 hours later, they could, because people who took LSD were still feeling the effects of LSD, and people who took psilocybin were no longer. But at the peak of the experiences were completely indistinguishable. Yeah. Other questions? Yes, Mark von Zastro.
Bryan, as usual, that was fantastic. A question that comes to my mind with putting together the beautiful polypharmacology that your lab characterized, and then the distribution of the 5-HT2A receptor, makes me think that with some of the current transcriptional data that we have, there are a lot of GQ-coupled receptors that are expressed in these cortical regions. Yes. So do you think there's something special about the 5-HT2A receptor with respect to these psychoactive experiences? you think there's something special about the anatomical distribution of the receptors in the what explains the selectivity? Because I'm not aware, as you pointed out at the beginning, you know, the psychedelic experience is really specific to 5-HT2A.
It is completely specific to 5-HT2A, and there are some selective drugs, the N-bones, that are out there on the illicit market, which are extremely potent psychedelic agents. So I would say there's no question that it is the 5-HT2A receptor, for better or worse. I think it's all about the neuronal localization. So it's in these apical dendrites, it causes them to fire in this asynchronous fashion, and this basically mucks up a lot of stuff that's going on in terms of how we interpret the outside world. One of the things is it tends to shut down the ability of extraneous stimuli, many types of extraneous stimuli, to be paid attention to by the brain. So the brain basically pays attention to internally derived stimuli, which then it attaches meaning to, which is, to me, is really interesting. That this is unusual. So these other psychedelic drugs or hallucinogens don't cause such a memorable experience. It seems to be very characteristic of LSD-like drugs. In some cases, people say it's a life-transforming experience. Okay, those of you who lived through the '60s, I think, would say it's not a life-transforming experience. It's a transiently transforming experience. But if you talk to sort of psychedelic enthusiasts, they'll wax on for hours about how significant this experience is. You don't see that with other drugs. So you don't go to conferences on marijuana where people talk about how their marijuana experience transformed their life or cured their PTSD. Okay, so there's something there. Yes.
The introduction of noise.
Yes.
As an explanation for the psychedelic experience, and then the arborization or increasing of the dendrites of the neuron as the
spines.
Spines, yeah. I mean, it feels like the first one is more likely for the psychedelic experience, but is the second one more? I don't know if you agree, but is the second one more connected with antidepressant effects?
That's the idea. Yeah. That's the current hypothesis.
Well, so then they are separable in that sense.
They're separable, yeah, but the signaling for the spinogenesis appears to be downstream of the alpha Q. So I think it's possible if you get so there are these weak partial agonists that we've gotten very interested in that might be a path forward, basically.
To disentangle them.
Yeah, to make compounds that are not psychedelic, but are therapeutic. Okay, I think that might be a way forward for those. Yes.
I have a question along the lines of Max a little bit. If you introduced a GQ dread into the population that expresses 5-HT2C, would you what do you think would happen?
So we can do better than that. We have a selective drug for the 5-HT2C receptor.
Any GQ receptor give that same functional effect in the same population of neurons?
Yeah. So we're doing that experiment now. So we have the mice, right? So we can so we've done the experiment in two ways, and we're still not done with it yet. So I can't give you a definitive answer. What we found was when we used the sort of typical GQ dread, it was not sorted to the apical dendritic compartment, okay, faithfully. There was some there, but it didn't look like the 5-HT2A receptor distribution. So what we did was we put the 2A C terminus on the GQ dread, and that pattern is identical to what I showed you here for the 5-HT2A receptor. And we didn't give CNO, we give this other drug we made, dechloroclozapine. Nothing happened to the mice, basically. So we think that's probably because we didn't transduce the right population of neurons. So we focused, there's been a lot of focus on what they call the MPFC, medial prefrontal cortex in mice. Mice don't have a prefrontal cortex, but there's a region that people call that. So our initial experiments were to infuse the virus in that region. And so we got very circumscribed transduction. So I don't think we transduced enough cells. So I'll just tell you the experiment we're doing now. So we have this constitutive Cree mouse, and we have PHP viruses. So these are viruses that can be administered peripherally, but can transduce large amounts of cortex. So we made these, and we're addressing that exact question right now. If you just activate GQ, you get the effects of psychedelics. And we'll see. I don't know. Yeah, we don't have that data yet. That's why, for those of you who use chemogenetics, that's the reason I invented the technology, was to understand how psychedelic drugs work. I did not invent the technology so people could write papers and get grants.
As an end user.
I invented it just for my own personal enjoyment. And then we never got around to doing the experiment. So now we have everything in place and we're doing the experiment. Yes.
As an end user of dreads, thank you.
Yeah. Sure. Yeah.
My question is, are there variants among in the human population, both intron versus and exons, that would then alter your psychedelic experience?
So there are variants, and we have published on them, and they all have a small effect on psychedelic drug affinity or potency. So I don't know. I have actually got, because of what I do, I get interesting emails, and I've gotten emails from people who say they've taken huge amounts of DMT and have had absolutely no effect. And they wanted to know if I could sequence their genome, but I can't. But if anyone here has an approved protocol to sequence genomes of people who take illegal drugs, then I'm happy to forward these names. There are a few of these interesting people that take these drugs and there's no effect. And it'd be great to sequence their DNA, but I can't. University wouldn't let me do it. So there you go. Yes.
So the startling clinical phenomenon that you reported is that you take some drug like psilocybin, and a year later, you have this profound psychological effect.
Yeah.
So how do you maintain that? What is your conception? I mean, you can talk about plasticity in the acute response and la-di-da-di-da,
Yeah.
but how is that maintained for a year?
Yeah, that's the thing I find most interesting is that. And I have no idea what the hell is going on.
Which means, I mean, so
I think
the credibility
it's really
is, it's so hard to imagine.
Yeah. Yeah, it's I don't know if anybody here has taken LSD, but people I've talked to who have taken LSD still, the experience is
you can
still
remember
memorable.
some startling experience,
Yes.
but to have the whole organization of your psyche, if you will, go from having been depressed with all the things that are associated with it, to clear a year later, it's pretty amazing. It's pretty astounding. So it's hard to imagine how that could be true.
Well, so I talked to lots of people, people that seem reasonable. Okay. And I've heard lots of stories like this. So I would say it's not common that it's not going to, I would predict it's not going to be a common outcome that people will take one dose of psilocybin and their depression will be cured. This is not going to be a common outcome, but they're going to have some effect. But I've talked to really rational people that I'll just give you two examples here. So I was contacted by a venture capitalist who had made all his money in the hedge fund industry, who had severe cluster headaches. And he went to the world's best neurologists. Nobody could do anything for his cluster headaches. And finally, somebody at MGH said, why don't you take magic mushrooms? And he did. And he had a pretty, as he said, a pretty intense psychedelic experience. His cluster headaches are gone. They're completely gone. You know, we have no idea what the hell is going on. You know, I think it's super interesting. I don't, you know, we've looked at the obvious things, you know, we've done a ton of RNA-seq and proteomics. We don't really see anything in mice that gives us any clue as to what the hell might be going on. But I think it's probably a pretty interesting thing. There's probably something really important there, hidden away. Yeah. All right. Well, if people want to go, just go.
Have you looked into if there's an endogenous population of serotonergic neurons that releases serotonin onto the apical dendrite where 5-HT2A is enriched?
Yeah, so that's another question: is where are the inputs? You know, are these inputs coming from a separate class of serotonin neurons? We don't know. We have the tools now; we can do the mapping, but we just don't know. Yeah.
Thank you.
We're happy. By the way, we're happy to share the mice with anybody that wants them. We've already given them out to lots of labs. So, yes, you're back.
Thank you so much for a wonderful talk, Bryan. I was wondering, you recently had the paper on non-hallucinogenic or non-psychedelic antidepressants acting on the 5-HT2A receptor.
Right.
And I was wondering what kind of the consensus was. Is it still that you get the antidepressant effects because of the GQ bias or the lack of beta arrestin, or what is it exactly?
Yeah, that's a really good question. I would say it's not entirely clear. So it turns out that you can get, in our hands, a compound that's arrestin biased or a compound that's GQ biased. And provided some caveats are taken, they're the same. Neither is psychedelic in mice, and both are ostensibly antidepressant in mice, and they last for a long time. So one dose lasts for a month in a mouse. Okay. And I'm thinking this is just because they're just tickling GQ signaling, basically. So I think that's the key. I finally come around to that. I was did not believe it for a long time, but I think it's probably just activating that receptor. It hardly, it has low affinity for serotonin. It doesn't hardly ever see serotonin except under extreme circumstances. But it turns out if you just activate it just a little bit, it's fine. At least in mice. Yeah.
Gotcha. Thank you so much.
Yeah. All right. Okay. Thank you. Thank you very much.